Hello, everyone, and welcome to today's webinar, Changes to USP 643, Total Organic Carbon from Pharmaceutical Grade Waters. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit beckman.com. Now, we encourage you to participate today by submitting as many questions as you have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. I'd now like to welcome our speaker, Tony Harrison, a Senior Marketing Manager at Beckman Coulter Life Sciences. Tony, welcome. You may now begin your presentation.
Thank you very much for that introduction. I appreciate it. And welcome to everybody to today's short presentation on the new chapter for the USP 643 on total organic carbon. My name is Tony Harrison. I am a Senior Manager at Beckman Coulter Life Sciences. So a revised version of the USP chapter on TOC, total organic carbon, the USP 643 chapter, it's been published already, but it will become official on the 1st of May this year, 2021. This presentation gives an overview of this new document and discusses the difference also in the System Suitability Test materials specified in USP 643 and those specified in the Japanese Pharmacopoeia for TOC. Now, total organic carbon is one of the four critical quality parameters defined by the Pharmacopoeias for bulk purified water, water for injection, water hemodialysis, and pure steam condensate, plus, of course, packaged sterile waters. The other three are conductivity, microbial colony forming units, and endotoxins. USP 643 points out that TOC is an indirect measurement of organic contamination, and that whilst TOC can be a food for microbes, encouraging their proliferation in water systems, there's no direct numerical correlation between TOC levels in a water system and microbial contamination levels. It goes on to state that TOC can be introduced to a bulk water system from the source water, from the purification and distribution systems, and from biofilm growing in the system. For packaged sterile water, an additional source of TOC can be the packaging itself. USP 643 states that TOC is a limit test, and that means that the levels of TOC in a sample must not breach the defined limit, which is the response of the TOC analyzer to a 500 ppb standard made up from USP traceable sucrose. So in other words, whatever the TOC result the analyzer gives in response to testing the USP traceable 500 ppb carbon sucrose becomes the maximum limit of TOC allowable in the water system. So the limit may not be a reported result of 500 ppb of TOC, and users should be aware of this and ensure that the measured TOC levels in their water systems does not exceed the limit reported by their TOC analyzer when analyzing the 500 ppb carbon USP traceable sucrose standard. Just as an example, if the recorded TOC result from the TOC analyzer in response to analyzing the 500 ppb sucrose is a TOC result of 450 ppb, then a reported TOC result from a water sample of 450 ppb becomes the maximum allowable level of TOC for their water sample, not 500. Now, USP 643 does not mandate the measurement technology that should be used to measure TOC, just that whatever is used should have a manufacturer's specified limit of detection of 0.05 milligrams per litre, which is equivalent to 0.05 ppm or 50 ppb. Now, that it should pass the test for suitability, the System Suitability Test. USP 643 states that the analyzer should measure the TOC by oxidizing the organic molecules to produce carbon dioxide, and then measure the carbon dioxide from the TOC to calculate the amount of TOC present in the water sample. It does not mandate on or offline instruments, but it goes on to state that any technology used to measure TOC in water must be capable of discriminating between the TOC and the inorganic carbon that may already be present in the sample, total inorganic carbon it's called, such as dissolved carbon dioxide that may be already present in the sample or bicarbonate. Now, this discrimination can be achieved either by purging the TIC from the sample before oxidizing the organics, or by measuring the TIC in the sample and then subtracting that from the final total organic carbon measurement made after all the organics have been oxidized to carbon dioxide, total carbon. So some analyzers measure TC and TIC, do the maths and calculate the TOC, and this is perfectly acceptable according to the USP. Now, when the TOC test was introduced to the United States Pharmacopoeia in 1998, it replaced several other tests, and the challenge was to ensure that instruments used for TOC analysis would be suitable for analyzing the range of organic compounds expected to be found in pharmaceutical-grade waters. The System Suitability Test was introduced as a method to verify that a TOC analyzer could analyze with equal efficiency a solution containing the relatively easy to oxidize substance sucrose, as well as a solution containing the longer chain and more difficult to oxidize substance benzoquinone. Both solutions must contain 500 ppb of carbon, and a TOC analyzer must analyze both 500 ppb solutions equally well and produce TOC results from both that are within plus or minus 15% of each other. Now, as stated earlier, the TOC test is a limit test with a maximum allowable level in bulk pharmaceutical grade waters of 500 ppb of carbon. Now, whatever the TOC result the analyzer gives in response to testing that USP traceable 500 ppb sucrose solution becomes the maximum limit of TOC allowable in the water system. So the limit may not be 500 ppb, and as I said earlier, users should be aware of this and ensure that the measured levels of TOC in their water does not exceed the limit response reported by their TOC analyzer when analyzing that 500 ppb sucrose standard. Now, this section here, the system suitability testing for packaged sterile water, this is where the substantive changes to USP 643 have occurred. In addition to passing the system suitability test with the same plus or minus 15% limit, it now allows for different concentrations of TOC depending on the container size for the packaged water. And this is called L1. And as long as the water sample being tested contains less TOC than L1, it is deemed to have passed the test and the test is complete. If, however, the reported TOC value from a water sample is higher than L1, but lower than the second defined higher limit L2, the user is required to determine exactly what organic compounds are present in the water using a suitable analytical procedure. If the quantity of every organic compound present measured individually is less than 0.2 milligrams per liter, which is less or equal to 0.2 ppm or less than equal to 200 ppb, then they are deemed not to be present in a quantity that may be harmful to patient safety. If, however, any individual organic compound is found to be present in quantities greater than 0.2 milligrams per liter, then that organic compound must be evaluated for their impact on patient safety at that reported concentration. If it's determined that there's no impact on patient safety, then the water sample meets the requirements, and again, the test is complete. Now, if, however, any organic compounds are identified as potentially harmful and are present in sufficient concentration to impact patient safety, then the water sample does not pass the test and fails. Additionally, if the TOC analysis from the water sample shows that the level is higher than the second limit L2, then again, the sample does not pass the test and there's no requirement to identify and quantify individual organic compounds. However, of course, the user may require them to do this as part of their root cause investigation, but it's not mandated in the Pharmacopoeia. So this is the different package, the water container volumes that the new USP chapter uses: less than equal to 5 ml, greater than 5 ml, and less than equal to 100 ml, and then greater than 100 ml. And you can see there's two limits for TOC: L1, L2, and limit L1 is the first one. As long as it's below that, then the pass is a pass. If it's higher than L1 but lower than L2, that's when you have to analyze to determine what organics are present and in what quantities, and are they harmful for patients. If not, you pass. Great. If, however, the test comes with a result higher than L2 in the first place, then it fails. And those maximum limits vary according to the container size, as you can see by the table. And just a point of interest, the L2 limits are actually 50% higher, if you look at the table, than those for the L1 limits. So I did mention I'd comment on the differences between the United States Pharmacopoeia and the Japanese Pharmacopoeia on TOC. Now, unlike USP 643 chapter, which is primarily aimed at pharmaceutical waters, the Japanese chapter on TOC is a more general chapter and includes also reference to TOC analyzers that are used for cleaning validation too. And that's a fundamental difference. So as the organic compounds found in cleaning rinsed waters may be more complex and difficult to oxidize molecules than those expected in pharmaceutical waters, the Japanese Pharmacopoeia chapter on TOC calls for the TOC analyzer to be challenged with a solution of the ionic surfactant SDBS. So it's typical, an ionic surfactant can be used in the cleaning process, so they expect you to be able to analyze SDBS. Now, another difference is that the Japanese chapter gives some guidance on calibration, which is not the case with USP 643. The Japanese chapter instructs the user to use KHP as a calibration solution. Now, KHP is a relatively easy to oxidize solution. It's very stable, and it's frequently used to calibrate general purpose laboratory TOC analyzers. In contrast, USP 643 does not specify how a TOC analyzer should be calibrated, nor what the calibration solution should be made from, but frequently users will calibrate their TOC analyzers used to measure TOC levels in bulk waters, water for injection, purified water, and they'll use calibration solutions made up from sucrose because that is the reference material specified by USP 643 to determine the TOC analyzer's maximum limit for TOC in bulk waters. So this is bringing me to the end of my presentation today, and in conclusion, I'd like to draw parallels between this new USP 643 chapter and the subvisible particle contamination in parenteral drugs testing, chapter USP 788, where for small volume containers, the pass-fail criteria is based on the total number of particles per container. And the change to the test for packaged sterile water in USP 643 is suggesting that the total amount of organic carbon per container is also a measure of patient safety, not just keeping TOC levels below a set concentration as it would be in bulk waters. It is also asking users to do further testing when certain levels of TOC are breached to identify each organic compound in the water to better understand the risk to patients. Now, this is a significant change, and it will hopefully bring improved confidence that sterile packaged water is safe for human use. So thank you for attending today. I'll now take any questions that you have.
And thank you, Tony, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's take a look. It looks like we already have some great questions coming in. Our first question from an audience member says, thank you for your presentation. Please explain one more time why the maximum level of TOC allowed for packaged sterile waters is so much higher than for purified water and water for injection.
Great. Thank you very much for your question, and thanks for attending. So just to go over that again, the USP chapter acknowledges that while sterile water is derived from either purified water or water for injection, it's most likely that there will be additional TOC contamination from the container. Now, that's either because the TOC is present in the container when it's filled, or another way it gets in there is because these waters tend to leach ions from the plastic or glass container because they're actually very aggressive reagents. And remember, sterile waters usually undergo some sort of final sterilization process step. So the presence of TOC or higher levels of TOC is unlikely to encourage bacterial growth because any bacteria should be either dead or sterilized.
Thank you so much. Our next question, please can you explain again why sucrose or benzoquinone were chosen for the System Suitability Test in USP 643?
Certainly. So this often confuses. So the TOC test, as I mentioned, it was introduced into the United States Pharmacopoeia in 1998, and it actually replaced several other tests. And as I mentioned, the USP committee were concerned to ensure that any TOC instrument used to perform these tests should be capable of successfully oxidizing the range of organic materials to be found in purified water and water for injection. So they devised a System Suitability Test, which challenges the TOC instrument with a relatively short chain, easy to oxidize sucrose solution, and a longer chain, more difficult to oxidize benzoquinone solution. Now, as long as the TOC instrument can oxidize both solutions equally well, then it's deemed to be suitable for testing TOC in purified water and water for injection.
Thank you so much, Tony. And we have one more question from our audience. Why does the Japanese Pharmacopoeia call for system suitability testing on SDBS only?
Good question. And as I said earlier, the scope for the Japanese Pharmacopoeia is broader than that of the USP chapter on TOC. And the Japanese one encompasses not only TOC testing for purified water and water for injection, but also cleaning validation. And the JP recognizes that laboratory TOC analyzers primarily are frequently used for both purified water, water for injection testing, and cleaning validation, where, first of all, much higher levels of TOC may be present, and much more complex organic molecules, such as cleaning solutions, may be present. So, in order to ensure that the TOC instrument can handle both applications, the JP challenges the TOC instrument to ensure that it can successfully oxidize and analyze the complex and difficult to oxidize SDBS solution. Now, as long as the analyzer can successfully recover more than 90% of the TOC from the SDBS solution, then the analyzer is deemed to be suitable for both purified water and water for injection testing, as well as cleaning validation.
Tony, I want to thank you again for your time today and for your important research. And I also want to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care and stay healthy. Bye-bye, everyone.